So in part one of the video, we talked about speciation, but we really need to address these reproductive barriers more. So as I said, speciation is the evolution of a new species. For this to occur, you must have reproductive isolation. And like I said earlier, there are a lot of ways this can occur. This can occur through something called allopatric speciation, meaning there's a geographic barrier creating this reproductive isolation, or sympatric speciation, meaning there's no reproductive barrier, but something else is preventing the populations from interbreeding. So this reproductive barrier, this isolation, can be due to allopatric or sympatric speciation. In allopatric speciation, take this population of fish. This area in the middle of the pond has dried up, and now this population of fish has been physically separated from this population of fish, and they're two separate lakes. So this physical separation has led to a geographic barrier, and they can no longer reproduce. So the reproductive barrier is a geographic isolation. So whenever a geographic barrier leads to speciation, this is called allopatric speciation. Sympatric speciation is when the reproductive barrier occurs without a geographic separation. This can be due to behavioral issues or temporal issues or chromosomal changes. And we'll explore those in just a second. So on your big sheet, you'll notice right here we have allopatric versus sympatric speciation. Allopatric speciation is due to some sort of geographic isolation. Sympatric speciation is isolation without a geographic barrier. But it's important to note that both of these result in reproductive isolation. You're preventing gene flow. So when we look at reproductive isolation, we can break it down into prezygotic versus postzygotic isolation. Prezygotic isolation is before the egg and the sperm unite. So prezygotic isolation is before fertilization and can include habitat, temporal, behavioral, mechanical, or gametic isolation. Postzygotic isolation is after fertilization has occurred and can include reduced hybrid viability, hybrid infertility, or hybrid breakdown. These usually occur further down in the separation. So these prezygotic methods and this geographic isolation is really what's probably going to cause this initial reproductive barrier. And then these postzygotic isolations are probably what's going to reinforce that separation in this hybridization issues of speciation. So if you look at this slide, you can see reproductive isolation. Up here are the prezygotic barriers, and down here are the postzygotic barriers. I highly encourage you to read chapter 24 in the book. It does an excellent job talking about speciation and really explaining these types of isolation. To give you a introductory idea of how these types of isolation work, we're going to go over the next couple of slides. So again, geographic isolation is this idea that species occur in different areas. For example, these two species of squirrel were separated by the two sides of the Grand Canyon, and this physical separation allowed a reproductive barrier to form, and over time they evolved into new species. Habitat isolation is a little bit different than geographic isolation. In geographic isolation, two populations are separated from each other. They are not part of the same habitat. In habitat isolation, two populations actually live in the same habitat, but occupy different parts of that habitat so they don't encounter each other. And that can lead to a reproductive barrier just because they don't encounter each other within the same habitat. For example, these two types of garter snake, they live in the same area, but one lives in the water and one lives on land, and so they don't encounter each other and they don't interbreed, creating a reproductive barrier. Temporal isolation is a type of reproductive isolation where two populations start to breed at different times of the day or different times of the year. For example, these two types of skunk live in the same area, so they are not geographically isolated. However, one mates in winter and one mates during a different time of the year, so they do not interbreed. So the reproductive barrier is a temporal barrier because they reproduce at different times in the year. Another type of prezygotic reproductive isolation or reproductive barrier is this behavioral isolation. For example, courtship displays. The blue-footed boobies mate only after a specific type of courtship display, and so if two populations use different dances or different courtship displays, they will not reproduce, and the reproductive barrier is thus a behavioral barrier. Another type of behavioral barrier is different songs, different courtship songs, like in these insects. Another type of reproductive isolation is mechanical isolation. Many insects have very specific shapes of their sexual organs, and if they don't fit together correctly, then fertilization cannot occur. So the reproductive barrier is the fact that they mechanically cannot fit together. Another type of prezygotic barrier would be gametic isolation. Maybe the sperm and the egg were able to join together. However, they didn't join together correctly. The sperm did not penetrate the egg. The sperm could not survive in the female reproductive tract. So it's again prezygotic. So all of the reproductive isolation mechanisms we talked about earlier prevented the sperm and the egg from joining together, meaning a zygote never formed. Sometimes, however, a zygote can form, but there are other issues after the zygote forms, and those are forms of postzygotic isolation. For example, reduced hybrid viability. That means that the hybrid or the offspring produced by these populations that are in the process of speciation don't live. Or they're infertile, meaning they cannot produce viable offspring. Or the offspring of the offspring are infertile. For example, there are many species of salamander in the specific genus that can interbreed, but their offspring are fragile and do not completely develop. Sometimes the offspring are not fragile. They're very vigorous, but they're sterile. For example, horses and donkeys can mate and they can have mules. Mules are very strong. However, they are sterile because horses have 64 chromosomes and donkeys have 62 chromosomes. Mules have 63 chromosomes and cannot produce offspring of their own. Finally, the hybrids in the first generation may be fertile. They may be able to produce offspring, but their offspring might be weak, and this happens sometimes in cultivated rice. There has been a lot of debate over the rate of speciation. One idea is that speciation happens gradually over a long period of time. This assumes that big changes occur due to the accumulation of many small changes. Another idea behind the rate of speciation is this idea of punctuated equilibrium, meaning the rate of speciation is not constant. There are long periods of no change followed by rapid bursts of change. This could be due to sudden environmental changes. And one of the major proponents of this model is Stephen Jay Gould. Finally, it's important to note that evolution is not goal-oriented. An evolutionary trend does not mean that evolution is goal-oriented. Okay, surviving species do not represent the peak of perfection. They represent good enough. A great example of this is the panda bear. Panda bear's intestines process food